our video, wasn't it?
We are going to start class now. So today's topic is the phases of mitosis. So today's goal is to figure out the purpose of mitosis, understand what is occurring in each phase, and within that we want to learn what terms we will be learning and applying, and then compare and contrast the difference between mammal cells and plant cells in their mitotic phase. So as previously we have learned that interphase is not part of the mitotic phase. So in interphase, it is the longer phase that we have discussed before. Does anyone remember what G1 phase is? Brett.
Growth phase.
Great job. So what are the phases of mitotic? Anyone?
Are they up there? I can probably read that.
Adam?
Prophase, metaphase, anaphase, and telophase.
Great. So it seems like some of us kind of have an idea of what the phases are. And now we're going to have some more information on each phase. So what is mitosis? So maybe growing up you've learned that gesture of the mitotic phase through your hands. And if you haven't, we can learn it quickly. So with prophase, right here, you have all the chromosomes all mixed up. And then, you can do it too if you want. And then in metaphase, all the chromosomes are aligned. So you see how your fingers are aligned? And then in anaphase, the chromosomes start separating. And then in telophase, in some cases you have two. So why is mitosis important besides keeping cells living and growing? Can anyone give a reason why mitosis is important in a real life situation? Okay. So an example is a paper cut. So like a cut. So this is important, and this is why you want to learn what mitosis is. It's the cells in your body need to replicate and divide in order to heal that paper cut you might have gotten earlier today. And there are many more examples which we'll learn later on. So chromosome. As you know, the chromosome in the nucleus is all everywhere, jumbled up. And then the chromosomes will then become dense as the phases go on. And here are some labeled sections of the chromosome. You have a chromatid, the centromere, which is the center part. Then you have the short arm, the long arm, and then these two are sister chromatids because they are identical. Any questions? So the first phase we have is prophase. Prophase, you have here all the cells. This is just right after interphase, where the centrioles have replicated and are starting to divide. You have the nuclear envelope breaking down and the chromatids condensing into chromosomes. And the centrosomes are going to the opposite sides, as I said earlier. And then you have the microtubules starting to extend and form. So prometaphase is not necessarily a specific separate phase in mitosis. It's within prophase and metaphase. So here you still have the chromosomes condensing. The kinetochromes are starting to extend. And you have microtubules starting to attach to the centers. And as well, the centrosomes are starting to move at opposite ends. Any questions so far? Am I going too fast? Okay. So then we have metaphase, which is the most simplest part. It's basically when all the chromosomes are starting to line up in the center. And each chromatid is attached to an individual microtubule or spindle fiber on the opposite pole. Then we have anaphase. Here is when the chromosome becomes chromatids, and start to separate and go to each individual side. Then the non-kinetochores are starting to come in and push the cell apart to the other side. And the reason that is happening is because in telophase, the cell needs to start to elongate to separate. So here you start having the nuclear envelope starting to form, and you have the chromosomes unfolding and starting to go everywhere and become chromatins. Then you have the breakdown of the kinetochores or the microtubules, and it's starting to become smaller. Any questions? So here we have the last step that's not necessarily in the mitotic phase, but is still part of the whole cell cycle division. Here in animals, you'll see a cleavage furrow, which will end up separating the two cells to become separate. In plant cells, you'll have a hard cell plate that forms to divide two cells. And these two cells will be identical. There was no crossing over, which we'll learn in meiosis, and no exchange is going on. So they're identical, kind of like twins. Okay, so now we're going to get up and actually apply what we have learned. We'll need some people to be centrioles, which is what divided, and we'll have some people be chromosomes and the kinetochores. So come on, get up. You can pick whichever position you want, or I can tell you which one you want to be.
Oh, yeah.
Chromosome.
Okay, great job. Okay, so now let's go into prophase. So prophase, you have chromosomes condense and they become visible. And nuclear envelopes are breaking down, so this is your whole cell membrane here, okay? Great job. Then prometaphase, the chromosomes continue to condense, so they're more solid. And then we'll have the spindle starting to attach to the kinetochores. Okay, and then metaphase. Metaphase, you'll have the chromosomes aligned. There you go. You guys are in a straight line. Then we'll have the chromosomes are aligned already. So each sister chromatid is attached to a spindle fiber. So there you go. Kinetochores stretch out and grab onto those centromeres. And then now we have anaphase. Does anyone know what happens in anaphase?
We pull them apart.
There you go. So the chromosomes are starting to pull apart, and the kinetochores are what's happening to them. They're starting to break down. There you go.
Good.
And then telophase. The chromosomes arrive at the opposite end, and they're decondensing. So you're going back. There you go. And then we have the non-kinetochores pushing out the cell. So then creating a cleavage. So this is an animal cell. So this is now separating, and you two have become, what have you guys two become now?
Two different cells. But they're the same.
Great job. You guys can sit down now. Thank you. Okay, so on Friday we'll be doing a lab, and when we look through the microscopes, you'll have two to compare and see each phase actually live. So an onion root tip and a white fish. And then here you can see the cell plate. Any questions? So in order to leave, since we're already almost done, you have two options this time instead of actually solving a problem. So if you want to grab scratch paper and a pen. So option one, you have to create 3 questions involving the material we learned. And this actually might be seen in a quiz or a task that we will have soon on this topic. And then option 2, just write a brief summary of what you learned today. And if you didn't understand anything, that is fine too. Make sure you write that down, and then we can go over it at a later time or at tomorrow's lecture. So while you guys do that, I will play you a video.
Hey guys, want everyone's attention? We're going to go over what we did yesterday. We're going to go over the stages of mitosis. All right, everybody ready? All right, let's go. Interphase of cell division is longest part. Nuclear membranes and packets it starts. The cells growing, mitosis process happen?
It actually doesn't happen as fast as you think it is. There are some videos that shows you the whole process right away, but it takes a while, especially since interphase is the longer period of phase that prepares the cells in order to divide. So it's not as fast as you think it is, but it's fast enough